Hello, my name is Brian Duff. I am Technical Lead for the PSC portfolio. Welcome to PSC Foundations Module 7, Feeder-Based Culture of Human Pluripotent Stem Cells. After this module, you should be able to understand the history of feeder-based HPSC culture, describe the steps involved in preparing a feeder layer, understand the culture methodology involved in a human pluripotent stem cell feeder-dependent culture system, and outline the advantages and disadvantages of a feeder-based system. This module contains 5 sections. Section 1, the history of feeder-based HPSC culture. Section 2, the preparation of feeder cells. Section 3, HPSC culture on feeder layers. Section 4, advantages and disadvantages of feeder-based culture. And Section 5, the summary. Sidebar sections will highlight applications and other useful information related to the curriculum. Figure 1 here shows an image of a human pluripotent stem cell colony on a feeder layer. Section 1, the history of feeder-based HPSC culture. In 1998, James Thompson and his colleagues at the University of Wisconsin derived the first 5 human embryonic stem cell lines, designated H1, H7, H9, H13, and H14. They did this by culturing human embryos to the blastocyst stage in G1.2 and G2.2 media. G1 and G2 media were formulated around the levels of carbohydrates present in human oviduct and uterine fluids at the time when the embryo is present. G1 supports zygote to 8-cell stage, and G2 media will support subsequent growth to the blastocyst stage. Then the inner cell mass of the blastocyst was extracted and plated on inactivated mouse embryonic fibroblasts, or feeder layers, in an 80% DMEM, 20% FBS-based system, also containing glutamine, beta-mercaptoethanol, and non-essential amino acids. This work was pivotal as it was the first time that human embryonic stem cells were cultured in vitro. The work was greatly influenced by Evans and Kaufman, whose work in the 1980s pioneered the culture of mouse embryonic stem cells. Around the same time, Paul Price and colleagues at Life Technologies developed a serum replacement called knockout serum replacement, which over time became known as COSER, to reduce variability in the growth of mouse embryonic stem cells used in transgenic models. A few years later, while deriving the first clonal H9 cell lines, designated H9.1 and H9.2, Michael Amit, in collaboration with the Geron Corporation and James Thompson's lab in Wisconsin, decided to switch out FBS for COSER, again to help reduce variability. They noticed that when FBS is missing, the media needs to be supplemented with BFGF to maintain the PSCs in an undifferentiated state. It is important to note that the use of feeder layers is still required when using COSER. So at this time, fully defined conditions for the culture of HPSCs was still elusive. It wasn't until 2006 that Tennille Ludwig from the Thomson lab described a fully defined feeder-free system for the in vitro culture of HPSCs. This was a pivotal turning point. Now researchers had much more control over their culture environment. We had finally reached a point where huge sources of lot-to-lot variability such as FBS and feeders were removed from the culture system, which yielded much more controlled growth of HPSCs in the lab. This was also the genesis of our teaser family of media, and this topic will be covered in more depth in PSC Foundations 8. Section 2, Preparation of feeder cells. First off, why do we use feeders? Well, we need to provide an environment where the cells will be anchored to the culture dish in order to grow. We need something to mimic the extracellular matrix in vivo. Feeders or MEFs provide this. MEFs secrete extracellular attachment factors such as transforming growth factor beta or TGF beta, activin A, laminins, and vitronectin. They also provide additional growth factors and cytokines that support the growth and help maintain the pluripotency of HPSCs in culture. When we look at the workflow for a feeder-dependent HPSC culture, the first step is to prepare the feeder cells. Once that is done, we can seed HPSCs onto the feeder layer. Step 3 involves maintaining the HPSCs in culture on the feeders to allow them to grow until a time to, comes to passage them again. To support the next passage, more feeders will need to be prepared. The first step in the preparation of feeder cells is their isolation. Making fresh MEFs is technically difficult and requires the expense and upkeep of keeping pregnant mice. The procedure itself is not easy for beginners, as if different cell types are identifiable when growing the MEFs, such as bone cells, the entire MEF culture has to be discarded and the researcher must start again. We do not have any protocols or recommendations to provide customers for the isolation and preparation of MEFs from mouse embryonic tissue. Please suggest they purchase our frozen MEF products instead. A typical isolation protocol found in the literature is as follows. MEFs are derived from mouse embryos between days 12 and 14 of gestation. After day 14, embryogenesis is too advanced for making MEFs. The embryos are removed from the pregnant mouse. The head and red organs or visceral tissue is removed and the tissue is minced. The minced tissue is treated with trypsin EDTA and DNase 1 to create a single cell suspension, which is then plated in a gelatin-coated tissue culture treated flask. The cells are harvested 24 to 48 hours later and should be at least 95% confluent. The MEFs are usually expanded on gelatin-coated plates in serum containing high glucose DMEM. When they are 80 to 90% confluent, they are passaged. After about 3 to 4 passages, they are mitotically inactivated by irradiation or mitomycin C treatment. This is done to prevent fibroblast outgrowth while still ensuring they are live and able to secrete the necessary factors for HPSC attachment and growth. The inactivated MEFs, or IMEFs, can be used immediately or cryopreserved for later use. Section 3. HPSC culture on feeder layers. Now that the feeder layer has been prepared, you can seed the HPSCs. HPSCs can be seeded directly onto IMF-coated plates and maintained with daily medium changes. The media can vary based on the protocol. In feeder-dependent cultures, the Thomson lab used DMEM with select FBS. However, researchers who still use feeders more commonly use COSER supplemented with BFGF. The growth rate of the HPSCs will depend on many factors such as the media, the cell line characteristics, and the feeders used. It is important to note that all feeders are not equal and their performance is application dependent. Different strains of mice, different harvesting times and techniques, when and how they are mitotically inactivated could all affect how they will perform in an application. Therefore, it's very wise for researchers to qualify each feeder source for their application. When should you passage HPSCs on a feeder layer? Well, this should be done when any of the following occur. The IMF layer is 2 weeks old. 50% of the colonies are greater than 2,000 micrometers in diameter. Cultures have become too dense, i.e. greater than about 70% confluency. Or colonies exhibit increased differentiation. Panels A, B, and C here show HPSC colonies on IMFs at different confluencies. They should be passaged when they are about 70% confluent. If they have become overconfluent, they may begin to differentiate. So when it is time to passage, areas of differentiation should be marked and removed so that they do not carry forward to the next passage. The HPSC colonies can then be detached enzymatically using an enzyme such as collagenase 4 or mechanically by scoring around a colony, pushing back surrounding MEFs, and scoring the colony with a 22 gauge needle or pulled glass pipette. Once liberated from the MEF layer, the colonies can be triturated into smaller clumps and subcultured into new IMF-coated plates. Daily medium changes are then required until the cells are ready for the next passage. In figure 7 here, The culture has been treated with collagenase 4. The enzymatic treatment is complete when the edges of the colonies begin to curl and lift up from the plate. Section 4. Advantages and disadvantages of feeder-based culture. There are some advantages to feeder-based HPSC culture, although the majority of researchers now prefer using more defined feeder-free systems. Some advantages are as follows. There are well-established protocols and the original protocols for the in vitro culture of HPSCs on feeders. There is no requirement for a matrix to support attachment of the HPSCs. The required attachment factors will be provided by the feeder layer. The feeders will supply additional growth factors and cytokines to support the growth and maintain the pluripotency of HPSCs. However, there are disadvantages when using feeder-based HPSC systems. The isolation of MEFs is time-consuming and technically difficult. The researcher will need to maintain two cultures, the IMEFs and the HPSCs. These will require different media also. Areas of differentiation may be more difficult to distinguish in a feeder-based system, and therefore sometimes areas of differentiation can be brought forward to the next passage. In a feeder-based culture, enzymatic or mechanical passaging techniques are typically required, which are more stressful than gentler non-enzymatic methods. And finally, feeders are not defined and their performance can be variable. Therefore, researchers should validate the source of their feeders for their application. So in summary, inactivated mouse embryonic fibroblasts, or IMEFs, have been used to support the in vitro growth and attachment of mouse embryonic stem cells since the early 1980s. When James Thompson and colleagues derived the first human ES cell lines in 1998, they used an IMEF-based feeder system with serum-containing DMEM. Soon after, knockout serum replacement, or COSER, supplemented with BFGF, was developed as a way to reduce variability from different sera. IMEFs are helpful in HPSC culture as they provide extracellular attachment factors and growth factors and cytokines that support the growth and help maintain pluripotency of HPSCs. MEFs are isolated from 12 to 14-day post-coitum embryos. MEFs are expanded for 3 to 4 passages prior to mitotic inactivation using mitomycin C or irradiation. HPSCs cultured on IMEFs are usually passaged enzymatically or mechanically. And finally, IMEFs should be qualified for each application as there is variability between different MEF sources. Thank you for your attention.